and turn the presentation over to Shaylee.
Thank you, Dora. I'm Shelly Mahendra, and I'm really grateful for the opportunity to share my research through Microbial Insights webinar series. And I want to thank everybody for their time for listening in. So I'll be talking about biodegradation of 1,4-dioxane by a class of microorganisms. In this case, it will be specifically bacteria that express enzymes called monooxygenases. Just a quick background how I stumbled upon in this area. I'm a civil engineer by training and converted microbiologist, and I juggle some projects, Petri plates in my lab. So the main projects in my lab have to do with biotransformation of emerging contaminants, of which 1,4-dioxane and poly and perfluorinated compounds, among others, are some of the active projects. We also carry out molecular biological studies of a microbe that you'll be hearing a lot about during this webinar, Pseudonocardia dioxane-evorans. And my lab also looks at interactions of microbes with nanomaterials. In both ways, nanomaterials could be another generation of emerging contaminants, and we need to understand the environmental relevance of that. On the other hand, nanotechnology has the potential for environmental remediation applications, in addition to, or as an alternative to current existing technologies. So either way, it would be really important to understand how they interact with bacteria, which are at the foundation of all ecosystems on our planet. For today's talk, I'll be describing our research on 1,4-dioxane. I want to make that clear at the outset. I'll be talking about 1,4-dioxane, this compound on the left side, which is not the same thing as dioxin, although both are human carcinogens and priority pollutants and important environmental contaminants. So 1,4-dioxane would be the subject of today's talk, and that's what I do my research on. I don't really know a whole lot about dioxin biodegradation. So where does dioxane come from? Historically, the largest source of industrial use of dioxane is as its use as a stabilizer for chlorinated solvents, specifically 1,1,1-trichloroethane. Dioxane is also used in the manufacture of paper products and polyester processing, polyester producing industries. More relevant for the personal care products and other commercial products is dioxanes being found as a residue in makeup and lotions and shampoos and inkjet printer ink, coolants and fumigants, a whole bunch of common products. But recognize dioxane is a residue of the manufacturing process in these cases. So there's a lot of dioxane that's being used in industrial and commercial products around us. If we want to measure dioxane in a variety of samples, clinical samples, industrial samples, soil, water, other media, there are some standard available methods, and then they're summarized in this table. And I must acknowledge Everett Glover and Dora Chiang of AECOM for sharing this slide with me. One of the most common methods is the EPA 522, the bottom row in this table. In my lab, we measure dioxane primarily in water samples or microbial culture media, and we use a modified 8260 method. It's a gas chromatograph mass spectrometer-based method, because in most cases, dioxane is the only compound of interest rather than the suite of non-volatiles and semi-volatiles that these methods are optimized to detect and quantify. So we do know that dioxane is used in various products, and now we know that there are methods available to measure and very sensitive. and reliable methods are available now. So why does it all matter? Well, the thing is, dioxane is not really good for us. It's a probable human carcinogen, which means it's classified by the International Agency for Research on Cancer. Dioxane is also toxic to liver and our nervous system, kidneys, and low dose exposure to dioxane results in irritation of eyes, nose, and throat. So basically bad news. You don't want dioxane in your drinking water and in anything that you have to do with occupational settings or out in the environment. If you ever wanted to understand, know anything about dioxane and other solvent stabilizers, specifically from an environmental point of view, but were afraid to ask, I highly recommend this book that came out about 3 years ago, authored by Tom Moore and colleagues. It really has a comprehensive description of all environmental aspects associated with 1,4-dioxane, and it's a very brilliant textbook. Some data that Tom Moore was generous enough to share with me. These are very recent. About 2 months ago, results became available from UCMR3, I think, Unregulated Contaminant Monitoring Registry list for a detection of 1,4-dioxane in public supply wells. And samples were collected from a large number of these sources and sent to various commercial labs, all of which use EPA method 522 in detecting. And it turned out, really, the bottom line was that they found dioxane in nearly 13% of the PSWs that they sampled. And I remember it being suggested that other than disinfection byproducts, which are expected in drinking water supplies, this is the most frequently occurring xenobiotic in drinking water. So going forward, dioxane will become more relevant in terms of detection. We are finding dioxane in many places that we now start looking at. Also industrial uses. There are other studies ongoing for understanding toxicology better and correcting the cancer slope factors, other things, other non-lethal endpoints. And with all that happening around in the environment, given the specific chemistry and properties of dioxane, environmental professionals and engineers and regulators would have to pay attention to this compound, emerging compound that keeps emerging. Turns out we don't really have a fixed standard that everybody agrees on and is required to meet. There is no MCL in drinking water established at the federal level for 1,4-dioxane. Various states have their own regulatory health advisories, and they range from sub ppb up to about 85 micrograms per liter ppb. And if you can see, in the last 4 years, there have been steps taken by several states to revise those standards. And it's likely that going forward, there may be MCL established and the standards would become lower as more health effects and the occurrence of dioxane and more information becomes available for that. Again, I want to acknowledge Everett Glover and Dora Chang of ACOM for sharing this slide with me. So now we know we use dioxane, can't help it. It's not good for us. It's out in the environment. It's in our drinking water. What can we do about it? So some of the remediation options that are commonly used for removing organic compounds from water supplies are air stripping and activated carbon sorption. And they work really well for many organic compounds. But dioxane is extremely soluble in water, so it won't effectively partition into the air phase nor sorb to activated carbon or other surfaces to a significant degree that would allow us to approach or meet some of these guidelines. So these two technologies would not really work for dioxane. What does work is UV photolysis and advanced oxidation processes such as UV peroxide treatment, Fenton's reaction, things like that. They're very effective in completely destructing the dioxane molecule, but turns out they're very, very expensive in terms of energy requirements for the ex situ treatment itself and for pump and treat operations if those are required to treat groundwater plumes that are contaminated with dioxane. Since dioxane is so soluble in water, phytoremediation has been suggested and actually successfully tried in a few places as a treatment technology, but its application appears to be limited to shallow aquifers and the right conditions. Also, most plants do not break down the molecule itself, so it becomes a matter of moving a toxicant from one media, one place to another. And even though some dioxane is absorbed through the plant roots and then volatilized, evapotranspired through leaves, you may still end up with a toxic plant that then you have to deal with. So knowing all that technologies that are available and work for a lot of groundwater contamination, we started to explore bioremediation as a low-cost, effective, green, in situ option for 1,4-dioxane. And I'll take you through my journey of how I decided to work with 1,4-dioxane when I was a PhD student at Berkeley about a decade ago. And I've continued to work on this in this area, and it's been quite a ride, and I'm happy to share some of our successes, and hope you'll find some of the results of our research interesting and relevant to your own work projects. So early evidence about 2 decades ago, there were some reports that there were bacteria and fungi that were capable of removing dioxane from experimental cultures. There were also mixed communities, such as those found in activated sludge, that had been implicated. There's research coming out of Clemson and other places that suggested that dioxane was being biologically removed in these samples. And since then, there's been many reports, many papers of folks identifying various bacteria, some fungi, other microbes, and that have may be responsible for degrading dioxane, and I certainly contributed a bit to this area. But what we realized that not a whole lot was understood about what exactly are these kind of microbes, how do they do it, what is the mechanism, how fast does the whole process, how does it perform quantitatively? If dioxane is indeed degraded, then what is formed? And what would be the effect of other complex factors that are found in dioxane-contaminated real environments? So all those were not well characterized when I started working with this in about 2001. So since there was biodegradation, it was a good direction to start exploring, and that's what we wanted to do. So along with my PhD advisor, Lisa Alvarez-Cohen, well, first thing we did was okay, we need to change the limited number of partially available information of biodegradation. We isolated, characterized this microbe, called it Pseudonocardia dioxanivorans. The strain name is CB1190. I'll be referring to it as CB1190 throughout this talk. A whole bunch of microbial characterization. The important thing for environmental relevance is that not only does this bacterium use dioxane, not only does this break down dioxane, it uses it for its own growth. So it uses dioxane as the only source of carbon and energy. So the microbe thrives on it. In addition to dioxane, it also biodegrades 1,3-dioxane and benzene and toluene, tetrahydrofuran, it's another cyclic ether, diethyl ether, other ethers, some alcohols, not MTBE, but many other environmentally relevant organic compounds are being biodegraded by this strain right here. So I'll be presenting some more detailed studies that we've conducted using this strain. The next question we wanted to ask, so what about this strain? And maybe there are some cousins out there. What's common among microorganisms that would biodegrade, a carcinogenic compound like 1,4-dioxane. What's in it for them? So we went back to literature evaluating toxicity of dioxane. And it turns out in laboratory animal studies using mice and other smaller mammals, there are these enzymes in their livers. They are called cytochrome P450 enzymes, and they metabolize dioxane. And the metabolic products of dioxane then interact with DNA, and then that leads to mutations and then propagation of tumors and things like that. Turns out in many bacteria, aerobic bacteria, there are also these enzymes called monooxygenases that are very similar to mammalian P450s. So with this information, we formulated our hypothesis is that the same system that breaks down dioxane in mammalian systems could be the same system or similar system that would break down dioxane. And whereas it causes the problem in higher organisms, that just might be our solution direction for bioremediation of this compound in the environment. So we set about testing this hypothesis. Let me tell you a little bit about monooxygenase enzymes. They are multi-component, large complex proteins. They happen to have a really broad, many monooxygenases have broad substrate specificity, which means they can catalyze reactions starting with a large number of organic compounds as substrates. Monooxygenase enzymes react in a way they take molecular oxygen and they split it into activated oxygen radicals. One of those oxygen atoms is inserted indirectly into an organic compound that destabilizes the compound and then starts the degradation reaction. The other oxygen atom is reduced to water. So down here at the bottom of this slide, it's a generic reaction that is catalyzed by monooxygenase enzymes in which a reduced organic compound becomes slightly oxidized organic compound and is available for further reactions, and oxygen is reduced to water. It's a redox reaction. There are thousands of different types of oxygenases. There are monooxygenases and other dioxygenases, and which are among the class of oxidoreductase enzymes that are very common in living cells. Let me take the example of toluene to kind of describe the diversity of oxygenases that may be relevant for environmental contaminant degradation. So toluene is this aromatic benzene ring with a methyl group on the top. And it is acted upon by a variety of different monooxygenases, and all that's been established through a large body of work. We understand this very well. The monooxygenase is named based on which carbon atom does it attack and insert that oxygen atom into. If that initial reaction takes place on the methyl group, on the aliphatic chain, that's called the side chain monooxygenase. If the ring is hydroxylated, then depending on the position of carbon that's hydroxylated with respect to the methyl of toluene, that's how these monooxygenases are named. So from top second through the fourth, it's ortho, meta, para, or 2, 3, 4 monooxygenase, toluene monooxygenase. There are also a class of oxygenases called dioxygenases, in which they take both oxygen atoms and insert them on adjacent carbon atoms in an organic compound. And that almost always happens when the compound contains double bonds between carbon and carbon. So this is kind of the suite of oxygenases enzymes, and we think they are relevant for 1,4-dioxane degradation. So let me summarize some of the data that we collected early on as we wanted to establish monooxygenase involvement in dioxane degradation. So here's the table. It lists some names of bacteria in the left column. The names are not important. Almost all of these bacterial strains, they're lab-based strains, they grow on toluene. They induce for a specific kind of toluene oxygenase, like the ones described in the previous slide. And some of them were able to degrade dioxane after growth on toluene, and one of them didn't. In addition to toluene degrading bacteria, we also tested what good 25, 30 other strains, many of which that contain monooxygenases were able to degrade dioxane to various degrees. Methane oxidizers, propane oxidizers, other strains. We also tested some dioxygenase expressing bacteria, and they were not able to degrade dioxane. E. coli, which was grown on algae broth, was really not specifically inducing for toluene. Also, it does not have an inducible oxygenase, and that strain also was not able to degrade dioxane. So we got some positive indication that maybe toluene oxidizing bacteria may be able to degrade dioxane. But how? Bacteria have millions of enzymes. How do we know it's this monooxygenase and not something else? So to answer that question, we got help from Professor Tom Wood, who was then at Texas A&M University, and he created recombinant E. coli using the toluene monooxygenase genes, coding genes, from other strains. And we didn't do this work, and please don't try this at home. But what molecular biologists can do is they can take a strain, say KR1, isolate the T4MO gene sequence on the chromosome or plasmid, transfer it on a vector, which is a piece of DNA that is used to carrying other pieces of DNA back and forth between cells. And it inserted this plasmid vector, PBS, kanamycin resistant plasmid, into E. coli, which previously did not have T4MO. So the cell at the bottom is the mutant now on the right side, and now it contains toluene 4 monooxygenase. And similarly, many other strains were prepared that now acquired the toluene oxidizing or toluene monooxygenase capabilities. Here's that table again, the one that I showed some time ago. The top 4 rows are identical to the ones I showed, and instead of yes and no for dioxane, I now provide quantitative degradation rates in milligrams of dioxane degraded per hour, normalized by biomass. The bottom 4 set of recombinant E. coli are all grown on algae broth. They don't really need toluene for growth, but they express toluene monooxygenases sourced from the strains that are among the top 4 rows. And as we see that the recombinant E. coli follow the pattern of the source strains, toluene monooxygenase expressing source strains. Yes, the rates are a little lower. These things are never 100% efficient, but there is consistency in terms of ability to degrade dioxane. This is pretty strong evidence that monooxygenases are the enzymes that are probably degrading dioxane, catalyzing degradation of dioxane. Here's another way of showing some of the same data. This is T4MO and T2MO, 2 different toluene monooxygenases. I'm showing time on the x-axis, dioxane concentration on the y-axis for both plots. The red diamonds, filled diamonds, are the source strains, so Burkholderia, the Pseudomonas, Mendocina, which contain toluene monooxygenase, and after growth on toluene, they degraded dioxane in about 5 hours. T4MO and T2MO is a little slower, but I'll get there. The green filled triangles are E. coli that contain the same gene from respective toluene monooxygenase expressing microbes. See, the thing about monooxygenase enzymes is that they are severely inhibited if they're exposed to acetylene gas. So what we did, we took the same reaction culture bottles with the microbes and the dioxane and growth media, but exposed them temporarily to acetylene gas, and then we degassed acetylene. What we found that once the enzyme was exposed to acetylene short term, it completely lost ability to degrade dioxane. So that also lends evidence in supporting our hypothesis that it's probably the monooxygenase enzyme, and was consistent across various cultures. Acetylene-exposed cells did not degrade dioxane, and those that were not exposed to acetylene did degrade dioxane, the source strains or the recombinant E. coli. And then we had some abiotic controls in here. To summarize, we were able to show confirmation of monooxygenase involvement by using 4 independent lines of evidence, and they all converged, supporting the monooxygenase hypothesis. We grew the cells that induced for monooxygenases. There was no dioxane degradation in the cells that were exposed to acetylene, suggesting it's monooxygenase. Recombinant E. coli expressing toluene monooxygenases degraded dioxane, but wild-type E. coli did not. And also we developed a colorimetric assay. See, the same monooxygenase enzymes also oxidize naphthalene to a mixture of 1 and 2 naphthals. And these products then react with an azo dye to produce a purple color. So that was good indication that the enzymes present and active, and the appearance of purple colors was strongly correlated with the quantitative dioxane degradation that we measured in the lab. So we're pretty confident that we got the right enzyme, and that would be important to other researchers and environmental professionals who are trying to consider biological options for dioxane degradation or other emerging organic compound degradation. So at least something to start with. In biodegradation, there could be 2 types of processes. The same enzyme is induced by a substrate, and it catalyzes that reaction in which the substrate is converted to products, and somehow this process benefits the cell that's producing the enzyme. And this is called growth-supporting metabolism. The same enzyme sometimes can be fooled into catalyzing a reaction of a different organic compound into producing products that provide no benefit, such as carbon or energy, for the microbe itself. And over time, if these useless or possibly toxic products accumulate, the cell experiences stress. And this is called co-metabolism, or a non-growth supporting metabolism. Fortunately or unfortunately, it's the second metabolism that is employed by environmental engineers and then trying to degrade xenobiotic contaminants in environmental media. Just the same explanation visually presented, that Pseudonocardia dioxanivorans, CB1190, uses dioxane for growth, and that's why the growth rates are rapid and linear. Pseudomonas mendocina, it's a toluene-degrading organism. It grows on toluene, but it co-metabolizes dioxane. So that's why after some time, since dioxane does not provide any energy to the cell, the degradation rates start to flatten out in the absence of any supplemental energy-generating co-substrates. So that's all typical, that's classical. I just wanted to put it out in front of everyone so that just by looking at the shape of the curve, we are able to understand at a macro level of what might be going on. Okay, so this is great. Dioxane is a problem, and now we know some bacteria and the kind of enzymes that they have that would break it down. So what happens when dioxane is broken down? Turns out in almost all of the cultures that I tested in the lab, 1,4-dioxane was completely converted to CO2 and water. It was brilliant. It was amazing. This summarizes, many years of work stumbling around, but we were able to confirm mineralization of dioxane using radiolabeled dioxane, and also identify major intermediates. The thing is that it goes all the way to CO2, as I said earlier, and none of the toxic intermediates accumulate for any significant period of time, which is a good thing that the reaction is thermodynamically favorable overall, and it proceeds to completion. One of the limitations of this pathway is that we don't really have intermediates that we can use for detecting or for monitoring in environmental applications to provide evidence of biodegradation in the field. But there are other ways to get around that. We also found that it was very similar pathway for both growth supporting and co-metabolic degradation processes. So we're able to establish this dioxane degradation pathway. Now we all know if there's a contaminated site, it's probably not contaminated by one thing itself. It's usually a mixture out there. And dioxane is found associated with chlorinated solvent plumes. It has been used as a stabilizer for TCA, but it's also found with TCE just because various industries and the military alternated between using TCA and TCE in their vapor degreasing and other operations. So this curve, this pie chart at the bottom left was provided to us by Hunter Anderson, who published a paper showing co-occurrence of dioxane and TCE in groundwater, or at least at Air Force sites. And we found that large fraction of TCA and many sites contain these compounds concurrently. We also know from scientific research and papers that monooxygenase expressing bacteria can co-metabolize dioxane. I showed that some time ago, as well as a variety of chlorinated compounds, including chlorinated ethenes, methanes, benzenes, other compounds, halogenated compounds. So that could be interesting, that you could use monooxygenase expressing bacteria to solve both problems, but you must understand the interactions of co-occurrence of these contaminants. So we have recently started a startup project led by UCLA, and our co-PIs are AECOM and Oak Ridge National Lab, in which we are assessing the biodegradation potential of dioxane in the lab as well as in samples collected from contaminated sites in the presence of chlorinated solvents. We also think metals are important co-contaminants for dioxane, and the way monooxygenases work, they have a necessary requirement for certain trace metals to carry out the catalyzing reaction properly. So it's important to understand the effect of metals. We also want to look at dioxane degradation potential in areas that are following biological reductive dechlorination, and what happens, how the geochemistry may change, the daughter products of reductive dehalogenation, other changes in the environment. And we have another sort of project. I'm a small participant in the project led by Dave Adamson at GSI and groups from Rice and GeoCleanse, in which they are evaluating other technologies for treating chlorinated solvents and how they may impact 1,4-dioxane environmental occurrence and treatment options in the site. So I'm really looking forward to working on these exciting projects. But I do recognize that it's really important to understand that we may have the best dioxane degrader in the world, but if it's not able to perform efficiently under these realistic conditions, then that's something that we need to know sooner rather than later. After I graduated from Berkeley, I accepted a postdoctoral position with Pedro Alvarez at Rice University, where we continued 1,4-dioxane degradation work together. And this is one project in which we looked at dioxane degradation in Arctic groundwater samples. So the data that I'm presenting here was collected at 14 degrees Celsius, so not quite Arctic. There's other experiments, but this would be pretty relevant temperature for aquifers in much of the country. So it's an important thing to recognize that not only under lab optimized conditions like 30 degrees, dioxide degradation can occur under certain environmentally relevant conditions. So since this is real samples, notice the x-axis on time is now in weeks instead of minutes and hours. The dioxane concentration on the y-axis is down by a couple orders of magnitude. In this plot, the light green squares are microcosms that were augmented by CB1190. And we saw that dioxane was completely degraded or below detection in about 6 weeks. At 14 degrees, I think that was fantastic. In some of our research, we found that there's a cousin of CB1190, Pseudonocardia antarctica, that was isolated from Antarctica. So we figured it must be adapted for low temperature growth, but it was not previously reported that this strain could degrade dioxane. Well, we set up a microcosm. and augmented it with Pseudonocardia antarctica DBS5A1. And it was even better than CB1190. So in about 2 weeks, dioxane concentrations were reduced to below detection. So really, never give up on microbiology, and try to find the right condition, the combination of growth substrates and buffers and bioaugmentation cultures that may actually lead to really high rates, surprisingly high rates of dioxane degradation in microcosms. And those experiments are a little more representative of field conditions than some of the work that we did using pure cultures. But that was also important in establishing the fact that dioxane is degraded and understanding the biochemistry in depth. The group, I am no longer associated with the group at Rice University, but they're continuing to do fantastic work. In fact, they published a paper earlier this month, I think in Environmental Science and Technology, that they've detected monooxygenases in various zones of 1,4-dioxane contaminated groundwater plumes. So I urge you to check that out. Going back to work that we are doing here at UCLA, and this is a large project that we did in collaboration with UC Berkeley, UC Davis, a number of national labs. It was funded by Department of Energy Joint Genome Institute. I think Pseudonocardia dioxane aborans is the coolest microbe on Earth, so I submitted a proposal to sequence its whole genome. It was accepted, and JGI did the sequencing, and we worked in collaboration and aligned and assembled the various contigs and annotated to produce a Pseudonocardia dioxane aborans genome map. This was announced about 2 years ago. The first author was Chris Solis, who was then at Berkeley and is now assistant professor at Drexel. The details are not important, it's just used to demonstrate that we recognize DNA sequencing information can provide really useful information on the capabilities of a microbe and what it has potential to do and how we can use that for a variety of environmental and other applications. What we do in my group, we mine the genome. That's a lot of raw data. This is a particularly large bacterial genome, has about 7.2 megabase pairs. We select genes of interest and see if they can serve as biomarkers for biochemical properties that are of interest to us. So we select those biomarkers. We design primers and validate those biomarkers and show that they're physiologically related to some of the things that we observe in the lab. And then we test that using pure cultures in the lab, microcosms, and in collaboration with ACOM and Microbial Insights, do some field-based experiments. So in the bottom row from left to right, it's increasing site representative research that we do, but it's also increasing complexity. So we try to balance both aspects in order to produce the most interesting and most meaningful research. So if you haven't guessed already, we are really interested in monooxygenase enzymes because I think we've convinced ourselves and some others that they are responsible for catalyzing dioxane degradation in certain aerobic bacteria. Turns out CB1190's genome contains over 2,000 sequences that could be monooxygenases. So we selected some likely candidates, started small, selected genes for methane monooxygenase or genes similar to those reported for methane monooxygenase in many other strains, phenol hydroxylase, phenol monooxygenase, and tetrahydrofuran monooxygenase. And then we designed primers based on starting with the Pseudonocardia genome, but also composite, taking inputs from other monooxygenases in the database. In addition to monooxygenases, we first wanted to see if we could just predict the potential for dioxane degradation in an environmental sample based on who's in there. So we know that some of these approaches work very well, say for dihalococoides, Nocardia, and other dehalogenating bacteria. Their presence alone is a strong indicator that reductive dehalogenation would take place in those samples. So we surveyed the tree of life for dioxane degrading capability, and it turns out it was scattered all over the place. In the bacterial domain, there were gram-positive and gram-negative and proteobacteria and actinobacteria and all kinds of things that had been reported to degrade dioxane. So we couldn't really narrow it down to a particular clade and say, hey, if this microbe's present, I think it's a pretty strong chance that dioxane would be degraded. We can't say that based on just phylogenetic relationships, but we did consider that. And in addition to 16S ribosomal RNA, we designed primers for what we think B14-dioxane monooxygenase. They were much more conserved. This is a much smaller region of the tree. And it turns out that the dioxane and THF and other types of ether metabolizing and co-metabolizing activity is strongly conserved in this little pink, purple branch right in the middle of this tree. And there's some scatter among the phenol, methane, or toluene oxidizing bacteria and then monooxygenase sequences associated with that. So taking all this monooxygenase information into account, we designed primers, small pieces of DNA that help synthesize copies of template DNA in a thermal cycler in the lab, but inside living cells also. So we targeted 16S ribosomal RNA genes to get a sense of the identity of the organism, but more importantly, monooxygenases as functional targets for 1,4-dioxane degradation. And I wish I'd be able to share a lot more fun details of some of our really new and exciting results, but it's not quite published yet. So I'll be happy to share more information. But what's important to recognize, and this is a good starting point, I'm showing data in which we have time on the x-axis in days, gene abundance, so it's a log scale, copy numbers per liter on the primary y-axis, and dioxane concentrations on the secondary y-axis. So these black filled circles indicate dioxane degradation. Unlike previous experiments, this experiment was started with previously grown CB1190 cells that were not exposed to dioxane. So it took some time to induce for dioxane monooxygenases, and that's why we see a little bit of a lag, around 3 days for that. And then once the monooxygenase was synthesized, it rapidly catalyzed dioxane degradation. As you can see, by day 5, it's all gone, and there's no significant losses in the abiotic control. At the same time that the dioxane was being degraded, the copy numbers of 16S, which suggests the number of bacteria, as well as methane, phenol, and tetrahydrofuran dioxane monooxygenase, they increase about 3 or 4 orders of magnitude. So that suggests that there is some connection between the abundance of these genes and dioxane degradation in this strain. So I'm going to stop there, and I would be happy to share more details as soon as this paper comes out. So this is one direction in which our current research is progressing. We are trying to understand and select biomarkers that would indicate consistently dioxane degradation capability in unknown samples. In addition to the biomarkers work, and by the way, this is a study that is funded by Air Force Civil Engineer Center BAA program in collaboration with AECOM and Microbial Insights. We're also looking at CSIA, compound-specific isotope analyses, as an indicator of dioxane degradation. So we do our experiment, we expose bacteria to dioxane and monitor dioxane over time. At various times, we collect samples and measure the ratio of carbon-13 to carbon-12 in the not yet degraded dioxane. The theory behind CSIA is that when chemical reactions or biological reactions take place, they preferentially target the lighter isotopes, so the bonds between carbon-12 and carbon-12. As a result, in the residual organic compound, the heavier isotope builds up. And we have the methods to pretty accurately measure the heavier isotope to lighter isotope ratio. And mind you, these are all stable isotopes, so not radiolabeled dioxane. This is natural isotope ratio for carbon. And then we feed that into this equation right here at the bottom. It's called a Rayleigh equation, a very well described and accepted equation to describe enrichment during chemical or biological reactions. Here I'm showing some early results, and unfortunately this paper is in review, not published yet, but it'll give you a flavor of how as dioxane is degraded, so work backwards with me, move from right to left, and fraction of dioxane remaining behind on the x-axis. So it's 100% at the right end, and then as it's degraded. As dioxane is degraded, whether it's via metabolic or co-metabolic process, there is an increase in enrichment of carbon-13. And it's a small change. It's parts per thousand per mil. But it's statistically significant compared to undegraded dioxane. But using the CSIA technique, we were not able to distinguish between growth-supporting and co-metabolic mechanisms yet. So this is ongoing work, but this paper should be out soon, that we do have a signature, an enrichment factor, when 1,4-dioxane is degraded by aerobic bacteria. So I think that's all I'm going to say. I do want to thank my mentors, my advisors, Lisa Alvarez-Cohen at Berkeley and Pedro Alvarez at Rice, for introducing me to this area and training me in all the environmental engineering and microbiology skills. I have an outstanding group of students here at UCLA, Pirapong Pornmontong, that some of you may know as ICE. He leads the CSIA work and some of the other co-contaminant work in my lab. Philip Gedlang is a postdoc in my group. He is leading the Monooxygenase biomarkers projects. And we have a wonderful supporting cast of master students and undergraduates that actively intern in our labs, and they contribute to our research program. In addition to those fine folks, I do want to recognize the funding agencies, especially AFCAC and CERDUP. ACOM, DuPont help fund some of our early studies. Microbial Insight's been very generous. BP supported the Arctic work. ICE is supported by Royal Thai Fellowship. We have collaborators all over, including some of those not listed here, and it's been a fantastic experience. So I just want to leave you with a quick summary of what all the things that we do in lab and what it means for environmental remediation of dioxane. I like to think that we offer information on not just the microorganisms, but also enzymes for dioxane degradation, and they may help environmental professionals in selecting inducing substrates or bioaugmentation cultures. Also, being engineers, we provide quantitative data that would be good inputs for models and help design reactors and in situ bioremediation systems. Identification of the pathway and sequencing of genome and biomarkers and availability of carbon isotope signatures would help develop monitoring tools for dioxane degradation in the field. And when I first started this work, I always thought that the flow of information was one direction from lab into the field. So this arrow was unidirectional. And over time, and I have collaborated and interacted and learned from so many people, I truly do think it's a two-way process. We get a lot of information from the consultants out there, from people who have a better understanding of the field, and there is back and forth exchange of information from the lab to the field to the lab, and that helps advance the field or this research area or in a meaningful way. And I'm grateful for the opportunity and to collaborate and work with so many wonderful people on this compound that's of emerging concern, and we hope to be able to make a difference in our professional lifetimes. So with that, I'll stop here. I think we still have about 10 minutes. I'll be happy to answer any questions Thanks again for the opportunity, Microbial Insights, and for all of you for your time. Happy to hand over control to Dora Ogles and be available to answer questions. Thank you.
Thank you very much, Shaylee. That was a great webinar. I really enjoyed it. We do have quite a few questions. The first question is, what water conditions are detrimental to the sustainability of the CB1190?
So the question is, what conditions are detrimental or what conditions are favorable? When we were trying to characterize the microbial adaptation of this culture, it seems to be pretty, near neutral type of an organism. We tested its growth at a variety of temperatures, and we found that the highest growth rates and dioxane degradation rates were observed at 30 degrees Celsius in the lab. But that should mean that it wouldn't work at any other temperature. As we saw from our subsequent work, it was active even at 14 degrees. But really 34 or 30 degrees or so would be the high end. So the rates are obviously thermodynamically slower at lower temperatures, but the organism can survive lower rates, diverse dissolved oxygen conditions. It has relatively lower tolerance for pH ranges. I did test pH from 2 to 12, and really somewhere near neutral, 5 to 8 was best. I don't think the cell grew at all outside this range. CB1190 can tolerate about 4% salinity. It's obligate aerobic organism, but monooxygenase enzymes are microaerophilic, so a relatively narrower range of oxygen adaptation. And CB1190 can use a variety of carbon substrates. Of course, we would prefer that it eats the pollutant of our concern. It can utilize a diverse range of nitrogen and phosphorus nutrients. It can fix its own nitrogen or use nitrate or use ammonia or urea or a whole host of amino acids. So, usually a good mesophile, I would say, unless there's a specific question about any of these. But these are general environmental adaptation, the physiology of the strain.
Okay, great. What are the lowest concentrations of 1,4-dioxane that are necessary to induce the monooxygenase? And what happens to the strain? How does it perform when you're below those minimum substrate concentrations?
That's an excellent question. So in an enzyme biochemistry, we think that it's first order with respect to the primary substrate, so that's the dioxane in particular. So with higher concentrations of dioxane, the transformation rates would be higher. In my lab, I think we have tested as low as about 50 ppb of dioxane, and the cell can grow and degrade dioxane in repeated cycles. Not a whole lot happens at concentrations below that, unless there's an alternative source of monooxygenase inducing substrate. So I would say we still have studies ongoing in the lab, but mid ppbs, nearly 100 ppb would be a good concentration that would sustain the growth of this organism. But we are exploring even lower and very, very high concentrations. We're exploring the whole range.
Okay, great. Does the presence of chlorinated solvents have any effect on 1,4-dioxane degradation?
I showed some data, and that was part of my PhD work, and it was just published earlier this year in Chemosphere, in which we do show that the presence of 1,1,1-TCA, as well as its abiotic degradation product, 1,1-DCE, were somewhat inhibitory for dioxane degradation under both metabolic and co-metabolic regimes. Of course, that's not the last word in this area. We do have ongoing work currently as part of our CERTA project, in which we plan to take a closer look at much wider range of concentrations and also VOCs, including TCE and vinyl chloride, to be able to have a much more comprehensive understanding. So ongoing work, there seems to be some inhibition, but if we are able to identify the problem, then we can figure out ways to best get around it as we can.
Has there been any work done to evaluate the potential for treating anaerobic water using maybe an ex situ type treatment cell?
I'm not aware of a field study in which they have used ex situ treatment, but I do know Professor Friedman's group at Clemson investigated dioxane degradation under anaerobic condition, and from what I remember from last year's Battelle conference, that it was a combination of iron catalyzed reactions, abiotic reactions, and the microbes, but the mechanism wasn't fully understood. I have tried many times to understand for dioxane degradation in the absence of oxygen, and we have not been successful yet. It could still happen. We are continuing our efforts. I can tell you that CB1190 will grow in the absence of oxygen under nitrate reducing conditions, possibly sulfate reducing conditions, but it will not, at least our data collected so far, if it's monooxygenase catalyzed reaction, this enzyme requires oxygen for it to work. So there may be an alternative pathway in some anaerobic microbes that could lead to dioxane degradation, but it's probably not monooxygenase mediated.
Okay, I know you touched on this a little bit when we were talking about the minimum substrate concentrations, but do you see a toxic effect on the concentrations of 1,4-D? Is there a maximum concentration that would be a concern?
We have, some by design and some purely by accident. These things happen in the lab. Sometimes scientific research progresses by serendipity. We've measured the ability of CB1190 to degrade percent levels of dioxane in solution. Mind you, this is in the lab when dioxane was the only contaminant. I think we measured up to 1%, so a number of orders of magnitude above what's a typical groundwater concentration, say 100 or below 100 ppb. So this organism can handle both. We didn't see any toxic effect, substrate toxicity, due to dioxane in this organism. I haven't tested some of the other organisms, but dioxane evorans can happily handle multiple cycles of up to 1% dioxane.
Okay, great. Have you looked at CB1190's effect on other compounds like PFOs or on bis-2 chloroethyl ether?
Not yet. But I would suspect that if this organism has enzymes that can break ether bonds, carbon-oxygen bonds, then it may be able to transform other ethers, although not all ethers. MTBE specifically was not degraded by CB1190, but the compounds that you mentioned, maybe. I haven't tried that yet in the lab, and I'm not aware of any other groups that have done this yet.
Okay. What about stable isotope probing? Do you feel that that's a good method to use to assess biological activity with this compound?
I do think stable isotope probing is a strong technique. At low concentrations, it's a good tool to verify the presence of potential degradations. But it has limitations, like we can't use it to determine dioxane degradation rates, as the SIP traps, such as those provided by Microbial Insights and other microseeps and other sources, they're baited with carbon-13 labeled dioxane. So also, given the low concentrations of dioxane out there, it may not be enough alone to confirm dioxane degradation, M&A, or enhanced bioremediation, but it could be important in the present, in conjunction with other molecular tools. So it can verify that the potential degrading bacteria are present, but it can't unanimously prove that, yes, dioxane is being degraded.
Okay. Aside from analyzing the 1,4-dioxane concentrations and looking at these biological tools, is it possible to look for any type of daughter products to help support biodegradation?
Um, I did share in this webinar some of the compounds that we detected that were a part of the dioxane biodegradation pathway, but the, they were not accumulating. So it's actually a good thing that dioxane was completely mineralized to carbon dioxide without anything toxic remaining behind. So there are breakdown compounds such as beta hydroxyetoxyacetic acid, ethylene glycol, glycoxylic acid, oxalic acid. But even if they are temporarily accumulating, although the lifespan is just a matter of hours, and that too at high concentrations, they're not really listed on an EPA, VOC, or SVOC methods. So it may be difficult to go after them. I wouldn't count on detecting dioxane degradation intermediates um, as a way to prove degradation. I think other chemical determinants and some of the tools that we are developing would be far more useful.
Okay. So if we're thinking about bioaugmentation, if there's a CB1190 culture that's been grown on 1,4-D, will it continue to degrade 1,4-D even when it's introduced to a low concentration groundwater, like something that's less than 50 ppb?
That's our hypothesis. So interesting that this question's asked. We were just recently awarded AFCAC BAA project, and that is led by Rebecca Mora at AECOM. And UCLA are collaborators in which we will prepare the bioaugmentation cultures to do a site demonstration. And our lab experiments suggest that 50 ppb is pushing it. It might just be the lowest limit that would sustain growth of CB1190. But this strain has surprised us before in terms of other things, so we're fairly confident that it should work in the field. And as I said, we have this ongoing work, and look out for some early results from our team at next year's Battelle or other venues.
Okay, one final question. What's the mobility of the CB1190 in a saturated aquifer? Do you see that it tends to adhere to the soil, or does it flow with groundwater?
I have done very limited number of column studies of various media and permeability. It varies. CB1190, I would be the first to admit that it's an actinomycete. It grows as in filamentous growth, but it, unlike other actinomycetes or high gram-positive bacteria, it changes its morphology from very thin, powdery, filamentous cellular shape to more roundish, I guess, more spherical shape at various stages of growth. So this is, again, something that we are testing right now. I don't have the answer for you. We do think we can control certain conditions, and we have a list of things to test for in the lab. And that would be favorable for good distribution and transport of CB1190 in the subsurface. But I don't have any clear answers for you. We are just days into this project. So soon, any minute now.
Well, thank you, Shaylee. That was a wonderful presentation. I really enjoyed it, and I think I learned a lot even from the question and answer session. So we really appreciate you doing this for us twice today. We will have this webinar available on our website so that I'm sure you'll want to listen to this again. And if you have any additional questions, please send them to us at info@microbe.com. We'll get with Shaylee, get those answered, and get back to you. Again, Shaylee, thank you so much for your time today.
Thanks for the opportunity to reach out to so many people, and thank you everybody for listening in. And I hope to meet with you and really share more formal and final complete story results of some of our real exciting research that's coming out of our group. So um, thanks again for your interest.
Great. Thank you, everyone. Have a good day. Thank you.
Bye.
Bye.